We isolated and purified the plasmid DNA using the alkaline lysis method that involves 3 buffers: resuspension, denaturation, and neutralizing to extract the plasmid DNA from the E. coli cell membranes, taking 1.5 ml of culture in 2 different vials. We centrifuged the tubes at 10,000 rpm at 15 degrees Celsius. Then we carefully removed the supernatant. We added 150 ml of resuspension buffer to the vials, and then we vortexed the vials and centrifuged it. Next we added 200 ml of denaturing buffer and vortexed them and centrifuged them. After centrifugation, we added 150 ml of neutralizing buffer, and then we inverted it immediately for 5 to 6 times. Then we thawed for 5 minutes and centrifuged the tube. After centrifugation, we transferred the supernatant in new vials. Next we added phenol chloroform equal to the amount of supernatant and mixed it thoroughly to get 3 distinct layers. The vials were then centrifuged and the supernatant obtained was removed carefully. We added double amount of isopropanol, and then it was centrifuged again and the supernatant was discarded. Then 70% ethanol was added and the supernatant was discarded. We air dried the vials for 4 to 5 minutes and added nucleus-free water to the pellet. We measured 1 gram of agarose and dissolved it in 100 ml of TAE solution. After heating the mixture, we added 20 ml of ETBR and the mixture was poured to create a gel. The gel was then extracted and kept it in the electrophoresis casting plate. 20 ml of solution and 4 ml of loading dye was then loaded in the wells carefully. We then observed the gel under UV trans illuminator.
In this video, we will discuss restriction digestion of plasmid PUC19 by using EcoRI restriction enzyme. Let's proceed step by step through the experiment process conduct. Requirements of reaction component are as follows: nucleus-free water, 10x assay buffer, 100x BSA, PUC19, EcoRI enzyme. Take 1 microfuge tube and place it on beaker containing ice. First, add nucleus-free water of volume 41.5 ml, followed by adding 5 ml of 10x assay buffer. Later, add 100x BSA of volume 0.5 ml, then add plasmid, that is PUC19, of volume 2 ml. Then thoroughly add 1 ml of EcoRI enzyme. Then total volume of reaction mixture is 50 ml. Later, we pulse the tube with the help of centrifuge for few seconds. Incubate the sample for 1 hour in water bath. Adequate 20 ml from 50 ml in another microfuge tube. Take 20 ml tube and add 4 ml of gel loading dye. Sample was loaded in agarose gel for electrophoresis. Result was obtained after 1.5 hours electrophoresis by observing gel under UV trans illuminator.
In today's video, we'll learn about the double restriction digestion of PUC19 by Ssp1 and BamHI restriction enzyme. It's based on principle that restriction enzymes are molecular scissors which cleaves the plasmid DNA at specific site and forms fragment of plasmid DNA. Let's proceed step by step through process. Requirements of reaction component are as follows: nucleus-free water, 10x assay buffer, 100x BSA, PUC19, BamHI and Ssp1. Take 1.5 ml of microfuge tube and place it on ice. Add 11.8 ml of nucleus-free water. Add 10x assay buffer of volume 2 ml. Add 100x BSA solution of volume 0.2 ml. Later add 4 ml of PUC19 plasmid. Add 1 ml of BamHI restriction enzyme and add 1 ml of Ssp1 to vial. Centrifuge the sample for few seconds. Add 4 ml of gel loading dye in vial containing sample. Load the sample in well for agarose gel electrophoresis. Result is obtained after 1.5 hours of electrophoresis. Bands are visualized under UV trans illuminator. In this video, we'll learn about quantification and purification of DNA bands isolated from an agarose gel. Requirements is as silica beads, sodium iodide, elution buffer, that is nucleus-free water, and wash buffer. Take an autoclaved vial and note down the weight. Subsequently, cut the DNA from an agar gel under UV transilluminator with the help of surgical knife. Took dose DNA in vial and weighed it. Later, took sodium iodide solution and 2.5 times of DNA's weight was added in vial. Place the vial in water bath at 55 degrees Celsius for 10 minutes. After 10 minutes, vial was removed from the water bath. Observe the dissolution of agar. Later, 20 ml of silica bits were added. Centrifuge it at 10,000 rpm for 30 seconds. Remove the supernatant in another vial without disturbing the palate. 200 ml of washing buffer was added and centrifuged at 10,000 rpm for 30 seconds. Repeated the procedure for one more time. Vial was kept in dry water bath at 55 degrees Celsius for incubation to remove traces of washed buffer. 20 microliter nucleus-free water was added to the vial and simply vortex it. For quantification of DNA, UV spectroscopy was performed.
Blue-white screening. Blue-white screening is biological technique particularly in context of PUC-19, commonly used in plasmid and cloning experiment. This method detects recombination bacteria by leveraging the alpha complementation process as a recombinant marker. This technique lies in enzymatic activity of beta-galactosidase encoded by Z-alpha gene which metabolize lactose to galactose and alternatively can hydrolyze X-gal to 5-bromo-4-chloroindoxyl to produce blue pigmentation. First, take the culture in 2 tubes and centrifuge it at 4000 rpm for 10 minutes at 4 degrees Celsius. Then pellets were resuspended by adding 5 ml of CaCl2. Again centrifuge at 4000 rpm. Resuspend the pellet with 0.1 molar CaCl2 and glycerol, adding 800 ml in each tube. "100 ml of culture was collected in two 1.5 ml vials. After that, 5 ml ligated sample and 5 ml control with no plasmid added to separate 100 ml competent cells. The tubes were kept in water bath at 42 degrees Celsius for 90 minutes. After water bath, the tubes were immediately kept on ice for 10 minutes. After that, 800 ml of LB media was added in the both tubes. After adding of LB media, the tubes were kept in shaking incubator at 37 degrees Celsius for 45 minutes. Then pulse it with the help of double centrifuge. 100 ml of transformed and control cell were spread plated on X-gal IPTG lurea agar plates containing ampicillin. After spreading on the plates, incubate both plates in incubator for overnight. Results.
In today's video, we will discuss about GFP cloning. Cloning is the process of making multiple copies of a particular piece of the DNA. So it's mainly based upon three principles: ligation, transformation and visualization. Let's proceed step by step. Measure 1.5 gram of agar powder. Add 1.5 gram of agar powder in 100 ml of distilled water. Measure 12.5 gram of LB for 500 ml of distilled water. Mix and stir it well. Add 400 ml of LB to 6 gram of agar. Add 100 ml of LB to 1.5 gram of agar and autoclave it. Later, pour media on a petri plate and allow it to solidify. Let's set up a ligation reaction. The component required, such as nucleus-free water, vector DNA, insert DNA, 1x ligation buffer, and T4 DNA ligase. Let's quickly go through the process. Take a vial, add 11 ml of nucleus-free water, add 2 ml of vector DNA, add 4 ml of insert DNA, add 2 ml of 1x ligation buffer, add 1 ml of T4 DNA ligase. Vortex the vial and place it at 4 degrees Celsius for overnight. Keep the LB broth containing colonase flask on the shaker incubator at 37 degrees Celsius for overnight. Culture is transferred equally into 2 tubes, centrifuged at 6000 rpm for 10 minutes at 4 degrees Celsius. Discard the supernatant, add 1 ml of ice-cold solution of calcium chloride to the tubes. After, resuspend it with 8 ml of calcium chloride solution. Place the tubes on the ice for 20 minutes. Centrifuge for 20 minutes at 3500 rpm at 4 degrees Celsius. Discard the supernatant. Again, add 850 ml of calcium chloride. Aliquate it into 8 tubes of volume 800 ml each. Place the tubes on water bath for 10 minutes at 65 degrees Celsius. Add 1 ml of controlled DNA and keep it on ice. Keep the vials in water bath at 42 degrees Celsius for 2 minutes. Immediately keep the vials on ice after 2 minutes. Add 0.5 ml of LB broth in both tubes. Keep the vial in shaking incubator for an hour. Take 100 ml of ligated sample and spread it on LB plus ampicillin containing plate. Subsequently, take 100 ml of control sample and spread it on LB plus ampicillin containing plate. Keep both plates in incubator at 37 degrees Celsius for 24 hours, and after 24 hours, observe the plates under UV transmitter. Result obtained from UV transmitter.